Hey guys, today we're going to talk about the lavender tubes. We're going to talk about the different tube colors. We're going to start with the lavender tube. We're going to go through the colors, what they're for, the sample type. There's much that you guys need to know. I will tell you that you will see a lot of questions based on tube colors, like the anticoagulants, what sample types are you collecting, how many times do you invert, so on and so forth. Please make sure that you make you some note cards and learn your tube colors and the ingredients. This chapter 2, can be very challenging because it's a lot of information in it. So I do encourage you to start making note cards and learning these tube colors. So one thing when we talk about tube colors, what information does the color coding of the evacuated tube provide? So the color gives us a lot of information, okay? The color tells us if there's a presence of an anticoagulant. It tells us what type of sample is being checked, and it also tells us how many times we need to invert that tube. So, and there's a lot of different colors, so, we're going to be jogging our memory here. We're going to start by talking about the lavender tubes. It contains anticoagulants. The anticoagulants in this tube are the K2 or the potassium xylate, the K2 or the K3 ETA. That is the anticoagulant, which is the potassium xylate. It also has it's a whole blood sample. We are going to invert this tube 8 times. And I'm not sure in the previous video if we talked about the inversion, but I'm going to flip back for a second to one of the previous slides just to show you guys. This is your inversion. This equals 1 inversion. So you're holding that tube upright, then you point that tube down, and then you pull it back up to upright. Once it's back up and upright, that is 1 inversion. So when we're talking about our inversion times, and then that's a full inversion. What you see on this slide right here is a full inversion. Upright, point it down, and then back to the upright position. That's 1 full inversion. So back to the lavender tube, we're going to invert that tube 8 times. So you can use the guide that I just showed you, and you would invert 8 times to mix this. The primary test that's pulled from a lavender tube is the CBC. That's probably the most common that you're going to see. So when you're thinking a lavender tube, you can think CBC, and you can also think this is going to hematology. It's whole blood, so it's going to hematology. The anticoagulants and stuff in this tube inhibit the platelets from clumping, and it helps maintain the cellular integrity, and it does not interfere with staining. The lavender tubes, the CLS, the Clinical and Laboratory Standards Institute, recommends spray dry potassium 2 ETA for hematology tests. So we're not going to use this tube for a coagulation test. So if we're doing a coagulation study on a patient, and look back when we talked about the coagulation cascade, coagulation is the time it takes for your blood to clot. If we're looking at a coagulation study, we're not going to pull a lavender tube because it has the presence of an anticoagulant that keeps it from clumping. So just think about that when we're talking about our anticoagulants and what types of tests are being pulled here. If we have an underfilled ETA tube, the excess anticoagulant may shrink the red blood cells, and it may make the hematocrit level appear to be decreased. So we need to keep that. We need to take that into consideration as well. Next, we're going to talk about pink tubes. The pink tubes is spray coated with the K2 ETA, the potassium 2 ETA. It is a whole blood sample with an inversion time of 8. And it's designated for blood bank tests. So there's a special labeling required by the AABB. And what we mean by this is it's a blood bank test, meaning they're typing your blood. So this tube is designated especially for that, and there's going to be a special label to go on this tube. The white tube Notice here the specimen type is plasma. So we're taking it back. Remember we talked about whole blood versus serum versus plasma. So with the white tube, they're looking at the plasma. It does have an anticoagulant. It's got the Edta-gel in here. And we're going to invert this 8 times. And it's used for molecular diagnostic testing. Now we're going to talk about light blue tubes. These light blue tubes, they have an anticoagulant known as sodium citrate. These prevent coagulation by binding the calcium. This sodium citrate is the most common anticoagulant, so you may want to write that down. We're going to invert this tube 3 to 4 times. It's primarily used for the coagulation test, and it preserves the coagulation factors. The ratio of blood to liquid sodium citrate is 9 to 1. It's very important that this tube must be filled completely. These tubes require a full draw to prevent dilution of coagulation, and when we're drawing this on a coagulation test on patients with polycythemia or hematocrit is reading greater than 55%, we must decrease the amount of coagulant to obtain that 9 to 1 ratio. So when drawing coagulation tests on patients with polycythemia or hematocrit reading greater than 55, we must decrease the amount of coagulant to obtain that 9 to 1 ratio. So please keep that into consideration. Incompletely filled light blue tubes will be rejected. They'll send them back. So just know that. Overmixing light blue tubes can activate the platelets and produce an erroneous coagulation result. So we want to make sure that we don't overmix that. The dark blue tubes, these contain the soybean trypsin inhibitor and thrombin, and they're used to collect samples for fibrin degradation products. The specimen is plasma. It's a thrombin and soybean trypsin inhibitor. Let's go on to the black tube. The anticoagulant here is sodium citrate. The blood to liquid ratio is 4 to 1. It's a whole blood sample with an 8-time inversion, so you need to invert it 8 times. And it's used for the Westergren sedimentation rates, which is what we'll commonly see as the ESR. And it's a specially designated tube and holder. Now we're going to talk about the green tube. The anticoagulant here is heparin, combined with sodium lithium. The blood type is plasma with an 8-time inversion rate. It is a chemistry test. Heparin prevents clotting by inhibiting thrombin in the coagulation cascade, so it stops that thrombin. It stops that thrombin in that coagulation cascade, so that's how it works. So one important thing here we need to know about the green tube is it does have the heparin, and that's the anticoagulant that stops or inhibits that thrombin. We have light green tubes. The light green tube, this is a plasma separator tube. The anticoagulant is lithium heparin. The additive is a separation gel. The type is plasma. We're going to invert this 8 times, and it separates the plasma from the cells. If we see the presence of a clot, this is acceptable in red stopper tubes, okay? But in the, the presence of a clot is acceptable in a red stopper tube. But we're talking the light green tube here, and I'm not sure why I have that note written down. But let's flip back to the light green tube. I got myself off track, and I do apologize. It's ideal for potassium tests and other chemistry tests. The heparin prevents the release of the potassium, so that's why it's ideal for the potassium. The vacuette manufacturer equals green and yellow circle. And like I said, this tube contains a separation gel. So we need to know which of these tubes do and do not contain that separation gel. And I apologize, I got a little sidetracked on that slide. So the gray tube it has an anticoagulant of the potassium oxalate or the sodium eta. It's available without the anticoagulant. With this gray tube, the sodium fluoride is an antiglycolytic agent. Okay, so that's important to know that the sodium fluoride that's contained in this gray tube is an antiglycolytic agent. It also contains a glucose preservative. That's that antiglycolytic. That sodium fluoride is that antiglycolytic, and it has that glucose preservative. Sodium fluoride is not an anticoagulant. It's not an anticoagulant. If plasma is needed, for this, the anticoagulant must be present. So basically, the sodium fluoride in this gray tube preserves the glucose. Sodium fluoride also decreases the microbial growth, which could produce alcohol. All right, we're going to go to the royal blue tube. The royal blue tube is plain with a clot activator. It can have sodium heparin or K2 potassium 2 eta. It's a serum or plasma sample. It can be either or, serum or plasma. If it's plasma, we're going to invert it 8 times. And if it's serum, we're going to invert it 5 times. These are used for trace elements, toxicology, and nutrients. Looking at our tan tubes, the anticoagulant is the potassium eta. The specimen is plasma. We're going to invert it 8 times. Contains less than 0.1 milliliter of lead, and it's used for lead testing. So one of the big things we need to remember about this tan tube is the need for the lead testing. We're going to talk about yellow tubes. These are for the blood bank. It contains the acid citrate dextrose, ACD, and it preserves red blood cells. The blood bank is a special cellular study. It contains the human leukocyte antigen, which is HLA, and phenotyping and DNA testing and paternity testing can be used with these. The yellow stopper tubes contain the sodium polyethanol sulfate and are used for blood cultures. These can also be used for blood cultures. We're going to talk about the orange tubes. The orange tube has an additive of thrombin with gel, rapid serum, RST. That RST stands for rapid serum tubes. This is a serum sample, inverted 8 times. It will clot within 5 minutes, centrifuge 10 minutes at high speed. So we need to make sure that we put it in the centrifuge, and we need to centrifuge it for 10 minutes at a very high speed. The orange tubes is recommended for a stat test with the patient receiving anticoagulant therapy. So tossing out the medical terminology here, stat means within 30 minutes you must draw this blood. So if you receive an order to draw a lab stat, that means you have a 30-minute window to get that lab in and sent to the lab so the doctors can see the results. So this orange tube is recommended for that stat, S-T-A-T, test on a patient receiving that anticoagulant therapy. So we're going to stop right here. We'll continue with the other tubes on the next video. But if you have any questions, please let me know.